All right, just to recap from yesterday, if you start on page 62, our first quiz is actually going to be tomorrow, 11/17, and our second quiz is actually going to be on the 28th after Thanksgiving break. All right, so we started off talking about protein synthesis, which is when our body makes proteins. We know that proteins are responsible for every single thing in our body. First thing we talked about was the DNA strand. If you look at a part of a DNA, it's called a gene, and one gene makes one protein. What do we already know? What are the instructions for making proteins? All the instructions in our body is called DNA. Where are these instructions found in the cell? DNA is always found inside the nucleus. Where are proteins made? Proteins are made at the ribosomes. And what are the subunits of proteins? Amino acids. So that just lets us know that we're going to start off with the instructions inside the nucleus, and then we're going to move to the ribosomes and find some amino acids. All right, moving on to page 63. On page 63, we knew there were two steps in protein synthesis, transcription and translation. I always remember that transcription comes before translation because the C in transcription is before the L. In translation, like an alphabet, the C comes before the L. So excuse me. In transcription, we just summarized and said this is where we go from DNA to mRNA. And the M in mRNA stands for messenger. The DNA cannot leave the nucleus, so a messenger RNA comes to deliver the message. This happens, of course, inside of the nucleus. We go from our double-stranded DNA and we do transcription, gives us our single-stranded mRNA. We know that mRNA or RNA period does not have a T, so A goes to U, T and A, C with G, C with G, G with C, so on and so forth. Excuse me. And then we have our second step, translation, is where we go from mRNA to proteins. This happens at the ribosomes, because ribosomes make proteins. So we take our single-stranded mRNA and we can use our codon chart, or we can use the tRNA to find the amino acids. After we find the amino acids, we put them together and then we have created a protein. Then we went on to try to figure out the differences between RNA and DNA. And for one, we know that RNA is single-stranded, whereas DNA, we can see, has two sides, so DNA is double-stranded. Another thing that RNA, the sugar, is ribose, where in DNA, the sugar is deoxyribose. And then we looked at the nucleotide. This is still phosphate. This is our sugar, but this time it's called ribose. And then this is still our base. And in RNA, the bases are A, U, T, C, G. There is no T in RNA. And in DNA, we still have phosphate. The sugar this time is deoxyribose. And the base is still there. And the base is A, C, and G. All right, using our codon chart, we had to fill in this part, the bottom of page 64. And we also concluded one codon, which is 3 letters, one codon is used to make one amino acid. So you use 3 letters to make one amino acid. Multiple codons can be used to make any one amino acid. So some amino acids have multiple codons, which means that We can make the same proteins even if our codons get changed a little. And this is called a neutral mutation. Sorry, a neutral mutation. Because it has no effect on you. All right, then we did a little transcription practice on page 65, which I know you filled in. Some of us struggle with number 7, so let me do a little bit of that. So below are some DNA molecules. Draw the messenger RNA molecule that would be produced from the left hand strand of each DNA molecules. The first 3 have been done for you. So G goes to C. We're only using the left side. T goes to A. C goes to G. A goes to U. T goes to A. So on and so forth. Perfect. And then we moved on to page 66, where you had to complete the chart. T goes to A goes to U, C goes to G, so on and so forth. In number 9 on page 66, you had to label the following. And DNA strand will of course be this one because it has T in it. A gene is a part of a DNA, and I'll let you guys fill out the rest. This picture right here is very, very, very similar to the one that we drew on page 63. The first thing you should do is name all 7 molecules and then label where's the cell membrane, where's the nucleus, so on and so forth. You also have to do page 67. And page 68. If you have not completed, please stop here and complete all the way to page 68. All right, we're moving on to mutations on page 69. A mutation is simply a change in the DNA basis. So if your DNA is supposed to say AAA, something happens in your body where the bases switch, and maybe it says ATA. So now your instructions are just a little different. Mutations can be positive, so this change could give you some type of advantage. Mutations can be negative. For example, some mutations can give you cancer, or mutations can be neutral. As long as it makes the same amino acids, then your mutation has no effect on you. Every person has mutations. Every single person will have a mutation. There are 3 different types of mutations. You have deletion. And of course, deletion is when one or more nucleotide is deleted from the gene. So mutation may supposed to be AAA, and deletion happens when one or more bases deleted. So your mutation now is AA. The next one we have is insertion, and this is when one or more nucleotide is added into the gene. So your normal DNA could be AAA, and your mutation could be AATA. Or you have substitution when one nucleotide is replaced with another, kind of like in basketball. So you're supposed to have AAA, but you have ATA. You have a substitution. What I want you to do now is to go ahead and complete the practice and try it out. You should work all the way up to page 70 and then hop on Canvas and make sure you have a check marks for every single assignment from units 1 through 4.